Alright, now question 5, which is a bit more problematic than the previous 2 questions. Right, so the question talks about this experiment, right, in which the cells were arrested at a particular stage in the cell cycle, after which, right, this fluorescent probe is mixed in with the cells after they have been arrested at a particular cell cycle, right? And this is the image that you will see with the fluorescence, right? Because of the fluorescence probe, and the fluorescence are all found at the ends of the chromosomes. Yeah, right, so these chromosomes, right, belongs to the stage of the cell cycle at which the cell is, or the cells are arrested in, right? So therefore, right, looking at the shape of the chromosomes, the arrangement of the chromosomes, you should be able to pinpoint the stage at which the cells were arrested in, right? So the chromosomes would look like that the cells were in prophase, right, when they were arrested. Right, prophase, this is showing you prophase, right? The chromosomes are not aligned at the metaphase plate. The sister chromatids are not separating, or chromosomes are not separating, right, to opposite poles. Right, the chromosomes are highly condensed. You don't see bivalence, right, because you don't see pairing of the homologous chromosomes. Right, each chromosome exists on its own. Then you actually do see, if you compare the size, the position of the centromeres between the chromosomes, you actually see that homologous chromosomes are present. Yeah, so all these point to the only phase that is possible here would be prophase of mitosis lah. Right, so for those who mentioned prophase 1, which quite a number of you did, right, there's no bivalent formation. For those who mentioned prophase 2, yes, the chromosomes look the same, right? In prophase versus prophase 2, the chromosomes look the same, but there are no homologous pairs in prophase 2. Right, but you do see, or you can deduce that there are still the homologous pairs present in this diagram. Okay, so therefore not prophase 2 either. And then for those who put at metaphase and whatnot, right, then obviously not because of the arrangement of the chromosomes. Yeah. Okay, then next part is a bit badly done. So let me go back to figure 5.1 first, yeah, and the information on top as well. Right, so figure 5.1 shows you a fluorescent tag and this DNA sequence, right? Then for those who skip the information, it says here it involves the use of a fluorescent probe, not tag, probe, consisting of a short DNA sequence and a fluorescent tag. So fluorescent tag is part of the fluorescent probe. So fluorescent tag is part of the fluorescent probe, and figure 5.1 shows the fluorescent probe used. So figure 5.1 shows the fluorescent probe used. This whole thing is the fluorescent probe, right? The fluorescent tag is this structure here that is part of the probe, right? And there's also this short DNA sequence, right? So it is not like how some of you have misinterpreted, right? Because so many of you mentioned that the fluorescent probe or fluorescent tag binds to the 3' end of a DNA strand, like the long DNA strand in the chromosome. The fluorescent tag does not bind to the 3' end. It is already bound to this short DNA sequence, and together the short DNA sequence and the tag together is the probe, which then binds to your chromosome. The probe binds to the chromosome, not the tag binds to the 3' end of the DNA strands in the chromosomes. Okay? So how does then the probe binds to chromosome? Obviously through complementary base pairing with the DNA, the short DNA sequence, right? Not somehow, again, not somehow the tag itself go and bind to 3' end of the DNA strand, right? So there must be some complementary base pairing, right? There is this single-stranded short DNA sequence. And if you look at the sequence, right, and also look at where the fluorescence is, the fluorescence is found at the ends of chromosomes, right, which you know are where the telomeres are, right? And if you look at the sequence of the short DNA sequence on the probe, you would then be able to see that the short DNA sequence is complementary to the telomeric repeat sequence, right? So the fluorescence probe binds via complementary base pairing to the telomeres, to the telomeric repeats, okay? That's why the 0.1, yeah, binds to the telomeres via complementary base pairing, or is complementary to the telomeres, to the repetitive sequence of the telomeres. And if you recall, right, the telomeres has got this single-stranded portion, right? The single-stranded portion would be then able to form complementary base pairing with the single-stranded short DNA sequence on the probe. That's 0.2, yeah? And because this is in prophase, right, and each chromosome consists of 2 DNA molecules, or 2 sister chromatids, each of which is 1 DNA molecule. So each DNA molecule has got telomeres on both ends, right? So now we have 2 DNA molecules, so there are 2 telomeres at both ends. So the question says at each end of a chromosome. So at each end of a chromosome, there is 1 telomere from 1 DNA molecule and a second telomere from a second DNA molecule. Right, that's why the third point, yeah? So that's why there are 2 telomeric sequence, or 2 telomeres, which the probe can bind to on each end of 1 chromosome, right? 2 telomeres, each from 1 different DNA molecule or 1 different sister chromatid. Okay. Then to part B. Part B, right? Can you just use a ruler to draw a line up from the 70 years old part of the x-axis, and then draw a straight line to the y-axis to read the telomere length, all right? So if you did that, and if you drew the line with right angle, yeah, to the x-axis and y-axis, right, then you can read the telomere length more accurately. So it is going to be 6 kilobase for the leukocytes and 7 kilobase for the skin cells. Not 6.5, but it's 7, because it's between 6 and 8. Between 6 and 8 is 7, not 6.5. Okay? And it's for individuals of 70 years old only. Or the question is not asking you to compare the 2 lines or the changes in the 2 cells, like from 20 to 80 years old. It's only on one age, like 70 years old. Yeah. Then for part 2, right, cancer cells do not display the same trend. The trend is a decreasing trend, right, that you see on top. As the age gets older, right, the telomere length gets shorter. That's because, right, leukocytes and skin cells are both somatic cells, right? And if you don't remember, or if you didn't know, right, somatic cells don't express telomerase gene. Somatic cells don't produce telomerase. But cancer cells, if you look at the whole outline of cancer development under the cancer topic, one of the step in the multi-step development of cancer is the activation of telomerase. Right? So cancer cells do express telomerase. So therefore, right, they don't have a decreasing telomeres, right, because of the extension of the telomeres by the telomerase. Right, so how do cancer cells activate telomerase, right? So the cancer cells must be able to now start or turn on the expression of telomerase, right? So how do cancer cells turn on the expression of telomerase? Expression or no expression of a gene is controlled by the control elements, right? The promoter, the enhancer, the silencer, right? So therefore, right, to turn on the expression of telomerase, which usually it shouldn't have, right? There must be then mutation to specifically the control elements, which then turn on the expression of the telomerase. It's not mutations to the telomerase gene itself, right? Mutation to the gene itself doesn't turn its expression on. Mutation to the gene will affect the proteins produced if the gene is expressed, right? So to turn on expression, it should be mutation to the control elements, right? The promoter, the enhancer, or the silencer. Okay, so promoter, enhancer becomes constitutively active, right? Or the silencer becomes non-functional. Yeah. With that, then the telomerase will be expressed. The telomerase enzyme will be produced to extend the telomeres. And the significance would be that the telomeres will never reach the critical length to trigger apoptosis. So cancer cells wouldn't, right, have apoptosis, and they can continue dividing, right, without the outcome of the shortening of the telomeres to the critical length. Because as the cancer cells divide, the telomeres do get shortened, right? And replication problem always occurs. Okay, there's no stopping it. Yeah, but as the telomeres shorten a bit, the telomeres can be extended or elongated. So it will never reach the critical length, even though cancer cells keep dividing and keep replicating its DNA. Yeah. So that's the point. Yeah, that's the significance. Okay. All right, and last part, right? Right. So first of all, right, if you think about evolution and all, right, no species or no organisms choose to die early, right? So the short lifespan is not a reason for why the telomeres are shortening faster, right? So like some of you mentioned, because the mice have short lifespan, so the cells must die faster. So must have apoptosis earlier, so the telomeres are shortened faster. The short lifespan is a result of the fast shortening of the telomeres. It's not the reason for why telomeres are shortened fast. Okay, so your cause and effect is a bit strange, yeah? Right, so mice have a very fast shortening of telomeres. Right, telomeres are shortened when cells divide, right? When DNA is replicated. Right, so if the telomeres are shortened fast, then that really means that the cells divide faster, or there's more or faster, more frequent DNA replication. Right. So that's the most obvious reason. And for those who understand the mechanism of the end replication problem, right, you know that that's because of the inability to fill up, right, the gap left when the last RNA primer at the 5' end of the dotted strand is removed, right? So if the primer is longer, then there's a bigger gap, right? Then the end replication problem would be faster also. So that's why the second bullet, yeah? But not the lack of telomerase, because the question, as you see on top, right, is talking about the somatic cells from the different organisms, and none of the somatic cells in the different organisms express telomerase. So even for human, right, which shows such a slow, right, shortening of the ends of the chromosomes, our somatic cells don't express telomerase either. Right, so therefore, it's not the mice have got faster shortening, not because of the lack of telomerase rather. Yeah. Okay, and there is also no correlation, between organism size and genome size. Doesn't mean the mouse is the smallest organism, then therefore it has the smallest genome, and somehow smallest genome have got faster shortening of telomeres, which is also not true. I mean, if that's the case, then a human should not be slower than elephant, as you can see from the graph, right? Seagulls shouldn't be so similar in terms of rate of shortening to dolphin, but if size matters, or size of the organisms matter, yeah? So it doesn't make sense lah. Anyway, there's no correlation between genome size and organism size. Right, so yeah, just some of the not very correct suggestions from some of you lah. Okay, so anyway, so it's question 5, yeah?